Okay, we'll get started in just a moment. All right, so welcome everyone to the HNRCA Monday seminar. We have an exciting presentation today from our invited speaker and followed by a Q&A after the talk. You can type your questions into the Q&A box at any time during the presentation. We've also enabled the upvote feature in Zoom for your access. And we also have the chat open. And you can use the chat to talk amongst participants, or if you have any technical problem, that's also a place to put those questions or comments. So our speaker today is Dr. Diana Thomas, Professor of Mathematics at the U.S. Military Academy at West Point, with appointments at Columbia University, New York Obesity Research Center, and Pennington Biomedical Research Center. I recently had the pleasure of meeting Dr. Thomas this year. We had some back and forth regarding predictive models. That was through the center's involvement in the NIH Nutrition for Precision Health Initiative, NPH, of which Dr. Thomas is co-chair of the executive committee for the consortium and a PI for the Aiming Center, which is the artificial intelligence group. If you recall, that's the same group led by Dr. Bruce Wiley, one of our earlier seminar speakers. Outside of NPH, Dr. Thomas has extensive research expertise in exercise, fitness, nutrition, and body weight regulation using mathematical models and machine learning. Thank you so much for being with us today. I'm now pleased to introduce Dr. Diana Thomas.
Thank you, Catherine. Thank you for the invite, and thank you for having me here. I do want to highlight the background of this picture is actually taken from my cell phone on my morning run. That's how pretty it is at West Point. So a standing invitation to any of you that would like to come visit. We're, I think, about 4 hours away from Boston, so it's not a terrible drive. You do have to cross Connecticut. Modulo driving over Connecticut and the Merritt Parkway, it's not a terrible drive. So I want to thank my team. This is my NPH West Point side of the team that's you see, and yes, everybody is military except me, and that's because tenured faculty at West Point are military. I have an unusual position there in some ways. I like these two pictures of West Point because one of the things I like is that things do not change much at West Point. The first is a photo, I believe, from 1912-ish. Of cadets at the boards. We have boards that circle around the room. In fact, blackboards were invented at West Point. And the second picture is a 2015 picture, and it's not much different. I think this day they're wearing ACUs instead of their formal grays. So I like that I actually was a postdoc in the early 90s, the mid 90s at West Point, and then I came back in 2016, and I really delighted that nothing had changed, not too much. I have a great team I'm working with right now. You'll hear me mention their names. Dr. Gower has been working with us from UAB with her postdoc Cat Suite. Jonathan Krakoff and Emma Stinson. Emma just came up to West Point for a week. We had a wonderful, productive time together. And you'll hear Jake Laporte's name mentioned. He's a fantastic coder and mathematician. And Josephine Kamek is helping me keep Cadet Skylar Schaff in line and make sure that he's doing the right thing as a cadet project. So I'm going to start off with this study, which I know you can't raise your hands, but I think I usually ask people to raise their hand if they've heard the study, but maybe I should have asked people to not raise their hand if they haven't heard of the study. It came out in 2015 in Cell, and it was revolutionary in a lot of ways. And in fact, this particular study by Zevi et al. Was the study that was the start of our own Nutrition for Precision Health study. According to Chris Lynch, he looked at this paper and was immediately thinking about how they could scale this up into a larger, more diverse study. And here we are today. So it's a very inspiring study. And I'm going to just take you to the graphic abstract. which kind of gives you a picture of what that study is about. So in the study, there were lots of data collected. And the purpose behind that study is that we have data. We want to answer something about nutrition and responses to diets. But the way we've been doing it so far is we've kind of been looking at one thing, one input and one output. And so we look at those two things and we try to connect them maybe with a regression model or standard statistics and try to determine whether there's, through statistical inference, if there was something there with a hypothesis. Instead, what the ZB team did is they took data from 800 people in Israel and lots of data, not just one-to-one or a few primary and secondary inputs. They took the microbiome data and they took blood tests, questionnaires, anthropometry, and food diaries, and they threw that into, I like that this is a black box. They threw it literally into a black box to predict glycemic responses to standardized diets. And you know, what's really talked about this paper is the machine learning model. The machine learning model is great, spectacular, it predicts well, but really what was important about this is that it's not a statistical model, it's prediction. So it's predicting the heck out of this data, the best they can. And they have, in the paper, they really talk about how they've exceeded what's previously been predicted. So their Pearson correlation constants were higher. And so now they wanted to know why. For statistical models, you can use statistical inference and determine, you know, just think about regression. You can look at the coefficients and say, okay, what has happened here? It's explanatory. The black box had to be opened, and they used something called partial dependence plots to open the black box and find out why certain individuals predicted it in the way they did. And so that's what we call a discovery study. They're discovering new things in this data that they threw into this black box that led to the predictions they did. So it's a pretty exciting paper. Even this quick synopsis probably indicates that. So a discovery study doesn't mean you fish around in the data and hope for the best. That's not how it works. You do have to have an endpoint. You do have to have something you're predicting. And so what the ZB team did is they predicted postprandial glucose response curves, or PPGR is what I'm calling them. This is what they abbreviated it in the paper. And when I quickly say PPGR, it might not make sense, but that's what I'm talking about, is these response curves. Why did they choose that? Well, there's going to be an expected postprandial response to a meal, and you expect that to vary by individual. If anybody's taken an OGTT before, oral glucose test before, which if you've gone through pregnancy, you've taken one. The whole point of that is that some people will have a response which is going to be indicative of gestational diabetes, and some people won't. So they know that there's going to be expected response. And having a response that's high may be a risk factor for a whole host of diseases. Again, this is a discovery science project, so they didn't have a hypothesis. They were searching for factors that may explain these differences in response coming from an array of data. And this is quite different than what we might be used to, because typically heterogeneity is not our friend, because you want things to kind of point in one direction through a hypothesis test scenario. Here, heterogeneity is our friend. So we want to explain the heterogeneity. That's the whole goal. I had more questions about this, and Chris Lynch was great about answering these, because I went to him and I asked him, well, can you tell me what papers actually show that when you have a high response, I'm going to define high because mathematicians don't like using words like high without defining them. But can you tell me why that's a high response curve is indicative of a risk factor? And he said, you know, there really isn't evidence that the higher your response curve is, the worse off you are. But the whole idea comes from homeostasis. And Walter Bradford Cannon coined the term homeostasis. And really, it means that in our body, in the human body, we're trying to maintain conditions. You can think of body temperature as a good example. You know, when body temperature is higher than 98.6, you know something's up. And if it's 101, you know something's pretty bad because it's pretty high. If it's 102, you know it's worse. So the higher your body temperature is, the worse your condition of your body is, because you're not in homeostasis. It's the same idea applied to glycemic response, that if you deviate more than normal, which I'm going to define, it's a risk factor. It can't be anything good. So in the ZV study, the way they measure these glycemic response curves is they use a continuous glucose monitor. This is actually the picture of their continuous glucose monitor that they used. And then they gave individuals a standardized meal, and they blew up a 2-hour period after the standardized meal. So that's what you see in this inset picture below. And the deviation would be that blue area, and they called it the area. It's a measurement of the deviation, actually, is the area under the curve. And it's actually, they call it incremental area under the curve. I used to wave my hands around, say it's actually the difference between two curves. As a mathematician, calculus 2, that's what it is. So instead of waving my hands around, which I did anyways, but instead of looking at my hands and asking, is this woman crazy? I'm really thinking of the whole area under the curve includes this red rectangle below. They're taking fasting glucose and making a rectangle and throwing that out. That's the incremental area under the curve. The blue area is the deviation from homeostasis after you throw out the red curve, which is fasting. So I actually saw these curves and this whole idea. I had read the ZV paper, as everybody had, when we were preparing our proposals for the Nutrition for Precision Health. It's clear that this was the inspiration for Nutrition for Precision Health. But I didn't focus on this IAUC until I think our kickoff meeting, where people were talking about the IAUC and the response to diets. And I asked my co-chair on the steering committee, what are these things? And she said, it's the response curve. Leanne Redmond, it's the response curve. And I said, well, why are you guys using IAUC? I mean, AUC at this point, I was just calling it area under the curve. Why are you guys using that? And she looked confused. And I said, but there's other things we could do. Like, why are you using that as a deviation? Everyone's using it. I started digging into the data. I have a whole file full of references that were given to me by my NPH colleagues. And I pulled up a few random ones, and every single one of them, you can see that they are using IAUC. Here's another one that's using AUC. It might be IAUC. I'm not sure. I haven't read into the paper. I just pulled it up, and you can see that they're using some kind of area under the curve. Area under the curve is the integral. If you remember from calculus, it's usually the second semester of calculus. You start talking about the Riemann integral, breaking things up into rectangles, and then letting the rectangle slip down to 0, taking the limit. That is the area under the curve. Integration is one of those things that we use to measure curves. Even the glycemic index uses incremental area under the curve. So everyone's using IAUC. I talked about calculus quite a bit already. We have a whole class on distilling curves. That's called calculus. That's what calculus 1 and 2 are all about. An area of the curve is only one of the things that we look at. If you recall from calculus, and this is an insulin concentration curve. It's a response curve as well. You don't need to just look at glucose. So insulin response curves from one of Barbara Gower's data, it doesn't look as nice and clean as the picture that you saw from the ZV paper. They might have taken a nice clean picture for their paper. The curves that we see in real people, looking at them, this is in a 6-hour time window after giving a liquid mixed meal tolerance test. This is what I asked Leanne Redmond, what do you see when you see this curve? Because this is what I see. This is what I see. I look at this curve and say, oh, look at that. That's the absolute max. There's other local mins and local maxes, also called relative mins and maxes. Hey, look, there's an inflection point. And look at that. The speed is really fast here. It's the fastest right here. Speed, you can think of a particle going along this curve. When it does a quick incline, that's a faster speed. You can think of it as driving your car to work. When you drive your car to work, sometimes you drive faster and sometimes you drive slower. You don't take an average over the whole time. You sometimes go 55 miles an hour, and sometimes you go 35 miles an hour. This is the fastest speed, the location of the fastest speed, and this is the max rate of decline. I was asked, I gave this talk a couple times, so I was asked, what is the inflection point? I'll remind you a couple times because it's a weird animal. The inflection point is when concavity changes in a curve. So it's when, in this case, I made a curve that's concave down. It's a curve that doesn't, a bucket that doesn't hold water, and it twists and it becomes a bucket that does hold water. And it gives you information about the structure of that curve. More than just, is it increasing or decreasing? In this curve that I've shown you here, the curve is always increasing. So it's beyond that. So we have actually seen inflection points mean something. I've seen it in abnormal fetal growth. If you have, the fetus is always growing. If there is an inflection point that's too early, you either could be a small for gestational age case, or you could unfortunately be one that doesn't make it to term. We've also seen it in plant growth. Plants, like the fetus, are always growing. But when there's a change in climate, the inflection point, there's an inflection point. You'll see that height growing at a constant rate, and then all of a sudden the height's growing at a different rate. So we do see it in plant growth and fetal growth. So here's the problem. Back last February, I started asking, can I get anything from calculus-based, these other calculus markers? Beyond IAUC, because I'm asking now people to buy into this idea that maybe we can learn something from the other shape properties beyond incremental area of the curve and relate them back to some kind of risk or outcomes. So for that, I needed someone who had data, and I needed someone who had some kind of outcome. So Barbara Gower, who is also MPI for the Nutrition for Precision Health group, she is down at UAB, and her and I began talking, and she said, I have data. And she has, this is the structure of her data. Her individuals are all overweight or have obesity. And she has a segment in the group that has reactive hypoglycemia. I'm going to define that in a second. She gave all her participants a standardized liquid meal test. And you can see the macronutrient breakdown of that in the purple box. And then she collected 6-hour response curves for glucose, insulin, C-peptide, GLP-1, ghrelin, PPY. I know how hard that is to collect now that I've hunted for more data. And I can tell you Barbara Gower absolutely rocks, and her data is amazing. So what is hypoglycemia? It's a condition where your blood sugar, your glucose level, is lower than the standard range. Reactive hypoglycemia is the condition where it occurs later after food intake. So you have a dip in your glucose level below normal ranges occurring between 2 to 5 hours after you had your meal. Having reactive hypoglycemia is a risk factor for metabolic disorders, for example, diabetes, above and beyond just hypoglycemia. So it's a risk factor for other things. Okay, so now let's do a switch in the story. Let me pause here and see if you have questions. I actually talked to Sai Das about this, that, you know, there's research on education where they know that in a remote learning environment, people can only pay attention for about 10 minutes, and then they have a dip in their attention span. So let's pause for a second and see if you have any questions. I can't see the chat, but I think Catherine can.
Yes, I don't see any questions in the question or the chat right now.
Okay, I've eliminated one because I always got asked what is an inflection point. So I've added that in now.
Yeah, and we'll have time at the end also where we can.
Yeah, this helps break it up a little bit. So if anybody has a question, they can ask it. And if you have a question, I'm moving on, but we can pause again in a few minutes. All right, so we have exceptional faculty at West Point. And Colonel Jake Laporte, that's the person on the left without the gun. The person on the right with the rifle is his daughter. And this is from our basic training. They were marching back. They get up around 2 in the morning and they march, do an 11-mile march. And that's for any basic training, by the way. Any Army basic training has an 11-mile march as part of the end of basic training. This one, though, ends up on the streets of West Point, and we all cheer on the sides. And she's exceptionally happy because most of the cadets by the end of the march are looking pretty sad and tired. But He's phenomenally fit too, and one of our best programmers. So I had tried for some time to get these calculus markers off of a Visual Basic application I was just doing in Excel, using the difference quotient, which I don't have to get into that. It's like not a good way to do it. And Jake and I talked, and we decided that the easiest way to do this, the best way to do this, is to interpolate the curve with something that you can actually find the first and second derivative for. If you remember, I know everyone has taken calculus, and it might be a long time ago, but the whole of calculus 1 is about the derivative, taking the first derivative, taking the second derivative, and then you set those babies equal to 0. That red line is where the curve, wherever the curve hits the red line, that's where the curve is 0. And for the first derivative, that tells you where the local maxes and mins are, the peaks and the naders. For the second derivative, it identifies the inflection points. So it tells you the timing of those inflection points, and then you have the X value for the inflection point, and then you can plug it back into the original curve and get the Y value. So it's all built on this interpolation model written in Python. And that was Jake Laporte that put that code together. What that code does, we labeled this the canoculator. What it does is it takes it generates any response curves, glucose, insulin. It doesn't care what you're throwing into it. And it doesn't care how many time points you have. I'm going to get to that in a second, because that's actually an interesting thing. If you have less frequency of measurements. And what it does after it interpolates that curve, it gives you all the peaks and naders and the timing of when they occur. It gives you max speeds, that the rate, concentration per minute, the max speed of decline, the absolute max, the highest peak, and the timing of the absolute max, the inflection point after the absolute max. I just picked one to start with, and the total number of peaks and valleys, the total number of critical points, and it dumps them out into an Excel file. So this is what you get out as output. I wanted to show you some more curves. They don't look all the same. So this is a PPY, a glucose postprandial curve. And not all of them look nice and pretty like the one from the ZV paper. Just to remind you, inflection points where the curve turns. So this is a concave down to concave up here that you're looking at here. This is a C-peptide postprandial curve. Max rate of incline, absolute max, inflection points, and that has a couple more bumps. It'll catch all those bumps, the canoculator will. A ghrelin postprandial curve, max rate of decline was right at the beginning, as expected. You just had the meal. Absolute max occurred somewhere here. There's an inflection point, max rate of incline. So these curves can look phenomenally different. And the next thing we did is we started doing some data exploration. So the first thing, we took some of the measurements that were in the Gower database, and we looked at things like, what do the correlations look like and distributions look like between the inflection points of, or the absolute max between the two? What do they look like? This is the inflection point that you're looking at. You can actually see this is a classic picture that they show in the introductory stats class when there is a difference between two things. So you can visually see that it looks like the inflection point for BMI with the non-hypoglycemic reactive hypoglycemic group and that reactive hypoglycemic group looks different. And in fact, there was a correlation between the inflection point and BMI in the non-reactive hypoglycemic group. So we're seeing some things already from data exploration that tell us kind of where to look. Then I had Skylar, who is my research cadet this semester. If you're at Obesity Week, you might have seen him in talks. He had to dress in uniform for the talks, the presentations that he visited. And Major Josephine Kamek sat down and made sure that he was doing all the right stuff. So we're kind of supervising, make sure that it is done correctly. We just asked him to run t-tests between all these calculus markers between the reactive hypoglycemic group and the non-reactive hypoglycemic group. So you see, like, so these, and we pulled out only the ones that had statistical significant difference. And inflection points showed up, max speed of decline showed up. This should be minutes per, the timing showed up when it happens. So for example, the max rate of decline is faster for the reactive hypoglycemic group. That makes sense, because if you have reactive hypoglycemia, you're supposed to go down. And so it's faster in that group. The time at the max rate of decline is earlier. That makes sense, because if you have reactive hypoglycemia, you're going to decline compared to everyone else, and probably at a faster time, at earlier time point. The inflection points are harder to understand. For the two cases I gave you already, for the fetal growth and the plant growth, we had to think about that. But the height at the turning point is lower in reactive hypoglycemic patients. That kind of makes sense. Barbara Gower is still thinking some of these things over, like what's the biological meaning? And IAUC showed up too, but we caught some more things. Insulin, similar kind of markers. C-peptide, similar markers. That's not a surprise, because glucose, insulin, and C-peptide are all married to each other. What's interesting is PYY, which is I call it the appetite suppressant hormone. I have to find a way to think about this biologically. And GLP-1, they actually did not have IAUC come in as a difference between the two. And understand what this means. If you're using IAUC to represent the response to the meals, that means that in the paper, when you write it up, you'll say there was no statistical difference between reactive hypoglycemia and non-reactive hypoglycemia for PYY. When there was, they're inflecting too late for the PYY, for the non-reactive hypoglycemic group, or the reactive hypoglycemic group. GLP-1 too, we didn't have any with GLP-1s, and a very interesting thing to look at these days with all the new drugs that are coming out, the obesity treatment drugs. And you can see that the height of the inflection point is too low for the reactive hypoglycemic group. What that means, I don't know. We still have to think about it. I'm going to pause here before I get into these pictures and ask if you have questions.
I don't see any questions in the Q&A right now.
Hopefully people aren't sleeping. That's usually what happens in calculus class. All right.
Since we're paused, can I ask you one question?
Sure.
I won't wait till the end. Do you model residual error in the curves, or are they perfectly fit to the data points in the pictures you've shown?
So we used cubic splines. So we're not a statistical fit.
Okay.
So yeah, so I think that so the way the cubic spline code works, cubic splines is used a lot in numerical analysis, and we actually pulled from the numerical analysis book, and numerical analysis is used to look at differential equations and solutions to differential equations and interpolate curves through them. So they don't typically have a statistical purpose in the numerical analysis book. The cubic splines algorithm that we pulled forces that when you have a cubic spline, you're really taking cubics and connecting all those dots with cubics. But at the cut points, you need it to not have a sharp joining point. If you have a sharp joining point at the cut points, it loses the derivative. And you need it to be smooth enough to have the second derivative as well. And so the algorithm we pulled focuses on the fact that we want a smooth curve so that we can take the first and second derivative. And this actually took us a few months. When I started doing this, I was interpolating I found out cubic splines was the one way to go. The cubic spline algorithm I was using it had a first derivative, but it lost the second derivative at the cut points. So I went to Jake. That's when I went to Jake and I said, oh, I can't get this to work. And he said, let me try it. I think there's more.
Yeah, questions. Great. Thank you. Yeah, we have a few more questions. Why don't we ask them now? So Larissa Constantino asks, what was the reason of measuring both insulin and C-peptide?
So this was Barbara Gower's data. So she was measuring it for some other study with a hypothesis testing piece. So the original study was, and I think these came out of several studies actually, her data did. So I'm not sure of the original reason. I do have the papers and I can send you those papers. And but you know, if it was in her data set and she measured it, we looked at it. So that's why we looked at it, was in her data set. So we looked at it. And Roger asked why IAUC is higher in the non-hypoglycemic group. So that's interesting. When people have reactive hypoglycemia, their curve is going to go down further and faster than people who don't. So their area under the curve is going to be smaller. So the non-hypoglycemic group is going to have a higher deviation. In fact, the first thing I think when I wrote my first code using just difference formulas, which is, it's the crudest way to do things. It's the way you learn how to take derivatives. It's a few days before you actually get the short way to take derivatives with using power rule and all those things. I'm probably bringing back some really interesting memories from college or high school. See you guys, but you use something called a difference quotient. And we are told in numerical analysis class that you shouldn't use the difference quotient. It's a highly unstable way to do things. But when I did that, I found negative numbers for some of the areas. And of course, if you have a negative number, it's because there's a portion of the curve that's below the x-axis. And that's when Barbara said, oh, that's my group that's got reactive hypoglycemia. They dip down below fasting rates. So they have a negative chunk.
Okay, great. I think we're caught up on questions. So if you want to continue.
All right. So another thing we did is we looked at just correlation heat maps. I looked at as continuous variables. I didn't, Skylar actually has made graphs, and I'll have him make a slides out of those graphs looking at glucose and ghrelin, for example. We could put those together. He's done that already. I just haven't put those into slides yet. But I just wanted to see, like, how does it look compared to age? And you can pull out which ones gave a signal, and you can see the glucose value at the max speed, the speed, actually the highest peak value, the inflection points are all correlated to age. Negative correlations are total number of critical points. And I asked Barbara about that. Why would the total, so that tells you that there's a lot of bumping around if there's a lot of critical points. And that means the older that you get, you have less bumping around. And she said it's probably because we become insulin resistant as we get older. Oh, I thought this was interesting. Our max speed of incline, how fast we're going up at the beginning, is lower in those who are older. BMI, similar kind of thing. You see that the highest peak value, glucose inflection value, IAUC doesn't really give a signal, too much of a signal here. So again, this is a case where you say, oh, there was no, if you're considering the response to diet being only IAUC, it's telling you that there is not a signal there. And so your paper might just say there was no correlation between the response to diet and BMI, which is not true. There is a correlation. It's just some other metric on that curve. The highest peak value, the glucose inflection point. My cadet came back and said, ma'am, the inflection points are showing something, because he was kind of upset initially that he had to do something with inflection points. Every time he asked me a question like, ma'am, what is inflection point? Well, you just took calculus. So why don't you for next week give us a lecture on what inflection points are? So he was really happy and delighted to see it show up. The timing of the absolute max was negative, had a negative correlation to BMI. That means the heavier you are, the earlier you're going to have that highest peak. So I wrote that down there. Higher BMI implies earlier max. Jonathan Krakoff said percent fat is a really important measure. She had measured percent fat, and you can see timing of the highest peak value is a positive correlation. So the more percent fat you have, the later the highest peak value occurs. And the timing of the inflection point is also at a higher timing point. What that means, I don't know, but I have found that the timing of these inflection points are really important. For example, if any of you work in pregnancy or fetal growth, you have most babies grow in a sigmoidal shape. And so you'll see like an initial exponential growth and then a leveling off. And the inflection point would be right where it goes from exponential growth to log growth. So that inflection point sometimes happens too early. And it means that the baby's not, it's going to be at risk for small for gestational age. Max speed of incline and decline have negative correlations to percent fat. So the more percent fat you have, the slower you're going to incline and the slower you're going to decline. This is, let's see how to get this out of insulin. Nothing surprising here. IAUC showed up for insulin as a positive correlation. Just about everything lit up. And in fact, Barbara did this first. We ran everything in the calculator. We threw the spreadsheet over to them. They looked at it first and they said, oh, I think her subject title was very exciting. A whole bunch of things lit up for insulin. So you see that most everything is a positive correlation. Again, the number of critical points is a negative correlation and the max speed of decline is a negative correlation to BMI. C-peptide is not as interesting to me because it's kind of tagging with insulin, but you can take a look at that and see what thoughts you have.
There's a question if percent fat is percent body fat.
Yes, percent body fat.
Thank you.
And this is C-peptide again for age. BMI, I'm showing you like, we looked at every single one of these. You're not getting a chance to look at them really closely. That should be inflection point value, not glucose inflection point value. If you're somebody that studies beta cell function, these will probably be more interpretable for you, or you're probably thinking it through like Barbara Gower did. Ghrelin, I think the hunger hormones are interesting. So ghrelin's inflection point value had a positive correlation to age. So the older you are, the higher the inflection point value is. That's the location of the inflection point. The absolute max, the highest peak was higher in ghrelin the older you are, and the max speed of decline also was higher. So there were only positive correlations. Roger, no, they don't, because I'm not taking one measurement and then like looking at them in the multiple comparisons. I'm looking at different measurements over, so these are different measurements over the same groups. So for example, if I looked at percent fat over 4 different groups, I'd have to correct. Somebody asked about the clinical relevance of doing analysis beyond IAUC. The reactive hypoglycemic examples I gave you show you the clinical relevance, like things that IAUC is not picking up. Anywhere it didn't pick it up, we're showing that something is different. But it also tells you more information. So for example, if you know that the highest speed is different, or that you're inflecting too early, those are predictors for having reactive hypoglycemia. Okay, let's see if I got everybody. I thought it was earlier. Olivia, I have to go back and look, but I believe that they inflect too early if they have reactive hypoglycemia. And it just makes sense that, to me, the way I think of it is reactive hypoglycemia, it's almost like they're downturning so quickly, and everything is happening earlier. So if you think of a cookie cutter of a beta cell function response, then all the things that should be happening are happening too early, and then you're dipping down too low. It's like a left shift and a downshift of the curve you're supposed to have. So it makes sense to me that they would have an earlier inflection point. Okay, ghrelin and PYY. Or here we still on ghrelin and BMI. This is interesting. The timing of the inflection point, and I have to do a shout out. It's a stronger correlation than IAUC. The timing has a positive correlation. So it's the higher your BMI, the later you inflect. The negative correlation is the actual value that you inflect at. You know, it makes me think we should really teach calculus differently, because a lot of times students will give you the X coordinate, but not the Y coordinate. And then I give them a half points for that. And they say, well, why'd you give me half points off? Like, you only gave me half the story. You gave me the X coordinate. That's the time when it happened. And the actual value of what happened at that time is the other part. It's not a satisfying response. You can see on their face, they don't like that response, but maybe with an application like this, they'll be able to see, hey, both of these values are really important. This is ghrelin and percent fat. The only positive correlation was the inflection point value. You see, I have a couple questions. Okay, I think I answered them both. And age and PYY, max speed of incline and max speed of decline are positive. That means that your PYY is inclining faster with age and declining faster with age, when it goes up and when it goes down. And the inflection point timing is further out. And IAUC also was a signal here. And positive correlations of PYY to BMI. Inflection point timing was a negative correlation, so it happened earlier for people with higher BMI. Total number of critical points, it was more bumpy for the higher BMI people, the PYY was. And GLP-1, again, GLP-1 is like in the nutrition and obesity research news all the time now. Timing of the inflection point showed up as a negative correlation to age. Inflection point value showed up as a positive correlation. Absolute max, and you see IAUC showed up here too. And here's BMI. So, conclusions. I think that initially when I wanted to do this, I didn't want to waste people's time. Am I going to show you something that different than IAUC? And the answer is yes. The postprandial response curve is characterized by much more. You get much more rich information than just IAUC. Those calculus curve-based shape properties can diagnose risk with physiological meaning. For example, I just laid out some of them beyond just IAUC, which is just the deviation, deviation between, it's a measure of deviation. And we can see these curve shape properties are all a function of age, BMI, and percent fat. We have requested the data from NIH, from Jonathan Krakoff's group. And if you want to know what's worse for paperwork with one federal institution, it's working with another federal institution. So it took us about 6 months to get through West Point's data sharing agreements. And then now we're getting through NIH's data sharing agreements. And I think we're very close. But last week, a few weeks ago, actually it was a week right before Thanksgiving, Emma Stinson came, and she saw something very interesting. She ran her data through the canoculator. So they had sampled at every 30 minutes. And we saw from Emma's results that sampling frequency impacted those results, because her max speed of incline was 1 at 1 minute for the majority of the participants. So she looked at that one, is there a mistake? And then it dawned on us that when I went back and looked at the Gower data set, they had an actual time somewhere within the first 30 minutes, they had that max rate of incline. And so I told her, I think what's happening is it's defaulting to just 1 minute because you don't have enough frequency of sampling. So Sai and I have talked about this, actually, the frequency of sampling, whether we should have 5 minutes, 10 minutes, and we just blanket, as the mathy people said, you got to sample as much as you can, because the interpolation of the curve is going to depend on the sampling. So what this told us just from Emma's data is that we need to sample more frequently in the first 30 minutes. Otherwise, you're not going to catch that initial rate of incline. Where are we going from here? Well, we have other studies. We have requested data from Predict. Sarah Berry's really interested in this. Sarah is going to rerun her papers with the calculus parameters. And Jonathan, I think we're right on the edge. Steve Smith attended my talk at Obesity Week, and he's going to share data, and he says he has a lot of actually meal test data. And of course, we're waiting for NPH data. And you might have data, and we'd love to join with you and share any response curve data that you might have. It doesn't have to be just liquid. It could be a meal test that you gave. I've been approached by a few people who've watched this talk and said, hey, could you do it for, let's say, chamber data? You can do this for any kind of curve data, interpolate the curve, and then start looking at calculus-based properties. And of course, we're trying to interpret the full biological meaning of all these parameters. There's a lot to dig into here. But we think we're at the more exciting part now. We already see that there's a signal. Now it's just playing detective and figuring out what it all means. So I am going to stop there and do Q&A.
All right, thank you very much, Dr. Thomas. That was an excellent presentation, and thanks for answering questions. Let's continue with the questions as I see more coming into the Q&A box. Should I read these? This first one's a little long from Sai.
I understand what Sai is saying.
Okay.
So Sai is asking about how will this help in understanding other things? Well, one of the questions I think I asked you was, is it the curve you're after? I asked a few of you guys saying, is it the curve you're after? You are. You're trying to take that whole curve and collapse it. And then my next question was, well, why area under the curve? It could be maxes and min. The shape of that curve is dictated by more than just area under the curve. And initially, when we talked to Jonathan Krakow, he was asking the same question you were asking, Sai. And we got off the call with him, and I turned to my team and I said, he doesn't get it. He doesn't understand what we're trying to do. So make him 3 or 4 different curves that all have the same IAUC, but really look different. Show him that you can have very different looking curves that have the same IAUC. So IAUC is a very, very coarse collapsing of the curve. So this is precision, right? This is what we're after. And we can do it with a few clicks on through Python, Jupyter Notebook. So Sai, does that answer you?
So I don't think she can speak, but she can respond in the chat. I guess there's a second follow-up, but I think that's just a clarification. So some might argue that the AUC is a good representation of it's a simple way to just give an overall sense of that curve, even though obviously you're losing some information compared to the separate parameters that you can look at. Do you think there's any validity to that argument that it does serve as a good overall measure?
I think some of those pictures you saw earlier where it didn't show up as a signal, but the other curve shape, you're going to miss something, right? Let me put it this way. Let's say you take a class and the class is graded as pass fail, and you did really well in the class, and your friend did kind of well, and your friend just barely pulled over the pass line, but you guys both got pass, and then some of your friends failed, right? So but it doesn't really tell you the granularity of how they passed and failed and how well they did. So is pass fail a good measure of how someone did? It's okay. Like, it tells you like these people passed, these people failed, but it doesn't really tell you, it's not granular enough to tell you how well they performed in the class and how much they might have retained information. I think IAUC is pass fail. and some of these other curve shape properties, in addition to IUC. I never think you should throw away something, right? You're just adding to that richness. And it's easy to get. It's not that hard to get. Dr. Leah has a question. Can the canoculator be used to predict curves for individuals in relationship to food intake? So you're asking whether food intake can be used. You can use any input. That's like the ZV model, right? Instead of having just IAUC as the primary endpoint, you can have all of these things as a primary endpoint. And, you know, you're looking at the curve, your actual, in fact, you'll see in papers, they'll say the response to diet. It's not actually, it's a distilled response to diet, right? So this is giving you more rich information about that curve. Somebody asked if I published this work. I haven't. You're hearing about it, but what I would like to do is I would like to add to this a little bit more. It's just, I feel like with Barbara's data, her data set's small. I think it's great. But to me, I would like to show that it works in other people. Like if I was a reviewer, I'd look at this and say, well, what about people who are normal weight? Does it signal anything? She had people who were all had obesity. What about people who have, you know, the best data is the one coming from NIDDK because they have follow-up data. They have people who had normal responses and then they followed them up and they got diabetes later. Now, if you can take those curve shape properties in their normal responses and show they were predictive of a follow-up, then you've found a subclinical way to identify risk before, you know, early.
Great. Eric Miller asks, is there any value in developing a joint model for some or all of the features being computed?
Eric, great question. So you can do a couple things. You can do, you know, every one of these endpoints separate. So you can think of a neural network. Inputs are microbiome, all these fantastic things. Then you have this big black box and it predicts inflection points. And then you throw all these things in black box and it predicts max value. Or you can have a vector output. So there's neural networks that you can do and it predicts a vector. So you can keep them all stacked up like a shish kebab and then have it predict the shish kebab. So remember, this is the endpoint. This is not the model. It's a way to distill that endpoint. Eric, does that answer you for your question?
Eric, you can respond either in the Q&A or the chat, and we'll see if you have any further follow-up. So can you talk about how this curve fitting might be applied to data from a continuous glucose monitor, for example? I think all of the data you showed were individual kind of blood draws, it looked like.
So she used clamp. It's exactly the same. So you would just take the Dexcom data and throw it, you know, take 2 hours after, take the sampling and just throw it in, and it doesn't care that it came from some other device.
So there's no fitting problems, because then once the number of time points basically goes to infinity, I guess I'm wondering if the fitting, if there'd be more trouble tuning parameters in that fitting algorithm, because it might try to be fitting all of these small.
No, so you're thinking of like a least squares type way of doing it. So the cubic splines is a little bit different than that. It tries to just fit a cubic in between every single one of them. So you're not going to have a least squares type issue. I think 5 minutes would be very good. That's what we have is the 5-minute intervals. 10 minutes would also be good. 5 minutes would be better. Just to let you know, like this, if you ever thought math professors were evil, we are. I actually used to put an inflection point between the grid. The Georgia Tech students had this HPs, and they could, you know, it could graph the and then they could use the trace button to find all the curve shape properties and then spit them out because it was all multiple choice. They had to fill in bubbles. And I would put an inflection point right between the grid points. So a lot of things can happen in between grid points. Of course, I purposely stuck that there so they'd miss it if they didn't do it by hand. I'm not sure if that was a good way to teach, but so that's the thing. Like the fear is that maybe the max increase happened, like for Emma's data, there was something happening between 0 to 30 minutes. She just didn't have it captured, whereas Barbara did, because she had more frequent sampling.
Okay, so I don't see any additional questions. Let me just take a final look here. If anyone has more questions. Otherwise, we can wrap up. Thank you so much, Dr. Thomas. That was great. Thank you for answering all those questions. We're going to conclude the seminar now. Thank you for everyone for attending, and we'll see you next week.
Thank you. Bye.
Bye.